Hello and welcome to our first actual video for lecture. So we are going to be talking about scientific method. So really it all starts with finding patterns, and that's all science is people wondering what's going on. Why is this doing this? Every time I drop my pencil it falls on the ground. Why? And so math and science, like I said, are very similar, and how we find things and how we make sure it's actually legit science is we go through the scientific method and we test things. Everything's peer-reviewed. Several people have to do this. We don't have one person randomly doing an experiment. They could have messed up their data and we all believe it. No, everything in science is tested a lot. So first, so you can think about it, we see the moon, and so phase of the moon or the eclipse. So why does this happen? It's something that happens regularly. What's going on? After you observe, you're going to lead into a testable question. That testable part is very important. So when we're doing things in class, you can't come up with experiments that you might be going into Mars. It's just not testable here. So you have to think of things that you would actually be able to test with the materials that we have. For example, you stay up all night. You observe that you feel jittery. You don't feel right. You think you can feel your heart beating in your chest. You look at your arms. Your blood vessels seem to be more visible than normal. It's a pattern. It's occurred before. What's going on? So you're wondering why. Again, the question, your heart rate was higher than normal. Why? And you think of some possible reasons. Is it Mountain Dew? Is it caffeine? Is it stress? Maybe you have a big test. Lack of sleep. You want to know why. And so you first come up with your question and see if you are right. So a testable question means it's accomplishable. For example, how does caffeine affect your heart rate? And your next step is a hypothesis. What do you think will happen and why? You want to make sure that you include if, then, and because. And it has to go with the question. You can't come up with random hypothesis. This is all goes in together. So for example, if I ingest, just as a reminder, our question is, how does caffeine affect your heart rate? Your hypothesis is, if I ingest more caffeine, then my heart rate will increase. And again, that was directly from the problem question, because it stimulates my brain, which speeds up my heart. So you have to, this is where background information comes from. So maybe you already know that and that's what you think. But a lot of times we have to go research that. You don't want to do experiments that everyone else has already done. Try and find something new. Try and figure out something new. And then we set up our experiment. So you have to have a procedure. You need materials. Be specific on this. You can't just say, get materials. What materials? What do I need? Heart rate measuring tool. So am I using finger? Am I using blood pressure cuff, which will get my heart rate? What am I using? Caffeine in different amounts. I would actually like you to say the amounts, how much I need. Because if I'm sitting down and doing this, I want to know. Quiet place, and then you need a list of steps. Procedure, you guys will see with lab reports, I'll actually call it material and methods. So you have your materials, you have your method, your list of how you're going to do this. So somebody else needs to be able to perform your experiment exactly how you do, which is difficult. So you have to make sure you're including everything, but we're all in high school. You can assume that somebody will understand how to walk and stuff like that. You don't have to get too humble. Use bullets and number steps. That's usually the easiest way. So first, find your heart rate at rest with no caffeine in your system. No other disruptions or disturbances can occur. Drink 100 milliliters. Notice we're using the amount. Don't just say drink Mountain Dew. How much? Because that's going to affect your experiment. And record the heart rate. Repeat with 200 milliliters, 300 milliliters, and record. Again, make sure you're going back to your hypothesis and problem question. You can't change, if you say you think it's the amount of caffeine that affects your heart rate, you can't now test Mountain Dew versus water versus juice. You're not testing different amounts of caffeine. With this one, you are. Do 3 trials for each amount. That's an important part too. Maybe one trial is messed up. You want to have 3 different trials to make sure, so you can get the average and see what's happening most of the time. You can see stuff I've highlighted is very important to make sure you know. So as you are coming up with your experiment, you want to make sure you're thinking of the independent variable. Independent is what you are controlling. This is the cause. So I control. I'm the scientist. I'm controlling it. Example, the amount of caffeine. That's what we're changing. Dependent variable. Depends on the independent. The dependent is also the results. Now, when I say that, that doesn't mean I want you to tell me that the dependent variable, the heart rate was this much at 100 milliliters caffeine. No, I want you to tell me the effect. So for this one, the effect on the amount of caffeine is the heart rate. Again, this goes in with your hypothesis. Your hypothesis should say both of them. If you remember, our original hypothesis was if I ingest more caffeine, then my heart rate will increase. So if I ingest more caffeine, that's my independent variable, my heart rate will increase. Dependent. You should always have it. Cause and effect. That should be right there with your hypothesis. You also need constants. A constant stays the same. That's what it means to be constant. These are all those things that you need to make sure you're controlling so that you're not doing more than one variable at a time. So your test subject, you can maybe have different test subjects, but you're going to have to do a lot of trials. The type of caffeine. I can't switch from Mountain Dew to coffee to Dr. Pepper. No, I'm staying the same. I'm just changing the amounts. Type of measuring tool, duration of rest. So everyone has to sleep the same because we're trying to keep it the same. Measuring tool, I can't have one person using a computer, another person using their finger. You're going to get some differences with there. Why do you guys think this is important? You should be thinking about how, again, you don't want to test more than one variable at a time, because let's say I have the different amounts of caffeine, 100, 200, 300 milliliters. I have one person who slept 2 hours, another person who slept 12. Heart rate's probably not going to be the same. I've had people do experiments where they're testing, I forget, it was heart rate due to something else, and they gave them caffeine before. Well, that's going to affect heart rate. So just think about how you're testing this. The other really important part is your control group, which is your standard of comparison. You don't add anything to it. You have to leave them alone, so that make sure that by freak of nature, that's not why their heart rate increased. So nothing's tested, and you always compare that to your experimental group. So for this one, I'd give them water, or maybe nothing, but probably water. They're actively drinking, but they're not getting caffeine. Your experimental group is the one you're changing. So this is the one that you gave caffeine to. So you should always try and get a control with your experiment. Again, about constants, make sure everything is the same. And like I said, otherwise you don't know which variable caused the change. So you don't want to have caffeine and stress. We're trying to just test caffeine. I already talked about multiple trials. It will increase the accuracy of your result the more that you have. If you do it once, something could have interfered. You do it several times, people are going to believe you. If I'm taking a new medication, I don't want to know that it was tested on 5 people. I'd like it to be tested on 5,000 people so that I know it's safe. Next step. So we've made our experiment, we've performed it. Now I want my results. You get observations. You have qualitative, which is a word. So I think of it as quality and quantity. Quality, and over here, that's going to describe things. What color? What's going on? Quantity, it gives me numbers. So how many? My heart rate, that's a number. Quantity. And so you should also have data tables, graphs, pictures, samples, whatever will enhance your experiment. Graphs are just a visual way to look at data. It's a lot easier to see. You need to make sure it has title, scale, data, axis labeled, so amount of caffeine versus heart rate. And you usually just use the averages, otherwise it gets pretty messy. The last part, and probably one of the most important, is the conclusion. This is the part that I know, if you're paying attention, if you actually know what's going on in your experiment, you'll be able to explain it here in the conclusion. You're going to restate your hypothesis and tell me if you support or refute it. Use numbers. Tell me why. So, you want to guess what's going on? What errors did you encounter? So if you had bad experimental design, so let's say you did put different parts of caffeine, that's not an error. That's, you messed up on your experiment. These are things like somebody didn't, stuff that you can't really control. I'm trying to think of one, but I can't really think. Stuff that you can't control. Who's right? So everything is published in science journals for other scientists to review. If it's going to be accepted, it's going to be tested over and over and over, and the same results can be found. Also, just on that who's right thing, just because your numbers don't match everyone else's, don't change them. It's not a really right or wrong. That's the data you got. Use that. So to put everything together, and this is kind of hard to see, but that says ask a question, do the experiment, collect the data. You should have your hypothesis before, sorry. And then explain the data, do more tests, revise the hypothesis if you need, and then down here it says well-tested and accepted hypothesis become scientific theories. Patterns are beginning to become law. Theories and laws are basically the same. However, what a theory is we may need to add more things. Laws are more about patterns. We usually have math, like gravity. Really, gravity is even considered a theory. But theories are well-tested explanation, have a broad set. It can have laws inside of it. They're both, it's not that laws are better than theories or theories are better than laws, or they're both up on the very top. The biggest difference is laws are meant to really describe more of a pattern. And a theory is usually actually more complex and dynamic. A law has a single action. And that will be it for our first video. I hope you guys enjoyed.